How many of you have ever heard of toxicology before you walked into this room? Well, very good, because I never heard of it until I was well into my graduate work. So a lot has changed in the last few years. I'm going to talk about the pharmaceutical development process and the role toxicology plays in that process. I'm going to give you an overview of the cost and time associated with new drug development. And yes, this is the traditional pharmaceutical development as opposed to the regenerative medicine, but believe me, it's just as expensive to develop these more traditional drugs. And then try to talk a little bit about some of the national initiatives to address these bottlenecks. I'm going to explain some of the shift in emphasis that's been going on more recently in early development. And then I'm going to give you a few examples of new technologies and opportunities for entrepreneurship. Okay, what is toxicology? A brief answer is it's the study of poisons. It's a sophisticated discipline that deals with poisons and their effects. It uses multiple lines of inquiry to assess potential deleterious effects of drugs and chemicals. It's used to predict the risk associated with and provides tools to monitor human exposure. You can look at toxicology. It essentially breaks down to three basic areas. The determinations of the presence of a poison. That's forensic toxicology. And I put up here that when most people hear about toxicology, they think about CSI, you know, and the toxicology. That's analytical toxicology, looking for the presence of a poison. There are other branches that study the mechanism of action of poisonous stuff. Okay, we know the material will cause heart damage. What is the molecular mechanism involved in that? And then there's the characterization of poisonous properties, if any, of a drug or a chemical before it enters commerce. And that's where I play. And here we use a little bit of both of these areas. You know, and there's two things I want to leave you with. One is the importance of the dose-response curve. Back in the early 1900s, we used to actually be able to weigh. Here's Dr. Wiley, the first head of the Food and Drug Administration, weighing toast gravimetrically to look for contaminants. Now we have instrumentation, it's not inexpensive, that can measure things down to femtogram per ml. Now, to give you some idea, for any of you guys that, or ladies for that matter, that might be in a party where people are perhaps smoking things they shouldn't be smoking, and you inhale some of that air, we can still detect that in your system 3, 4, 5 weeks later with this type of instrumentation. It doesn't have to show any effects. It's well below what is called the threshold level. What's next after this? I don't know. Maybe somebody here in the audience will come up with an even more sophisticated way to measure things. The other thing I want to leave you with is the dose-response curve, okay? We can measure it. We can measure a response, and then you can graph out, for example, the exposure, the amount in the bloodstream, whatever indication of exposure you have or you want, and then you can look at the effect. And you can do this either on a population level or you can do it on a continuous basis within a single organism. And so you can start off, you can dose the individual, and you begin to see an effect, and eventually you can't give any more to see the same effect, all right? And when I say that we can detect something in your system weeks after exposure, we can detect things way down here where you wouldn't be feeling any effects, and, you know, so be careful, you know, out there. Okay, so preferably what you'd like to see is, if you're a drug company, is you'd like to see a nice dose-response curve for the desired effects and for the side effects, the untoward effects, the less desirable effects. You want this curve shifted to the left. But you can see that there's always a little bit of overlap. You know, right here, you know, there's always going to be an opportunity for there to be a side effect. By the way, law of Paracelsus. All things are poison and nothing is without poison. Only the dose makes the poison, okay? So when somebody comes up to me and says, "Doc, is it okay if I take?" I said, "Well, how much are you going to take?" It's that, very simply put, one molecule, it won't be harmful to most species under most circumstances. Okay, the other concept I want to leave you with is target organ identification. In toxicology, the name of the game is often target organ. What is the target of the toxicity? Is it the same as the target of the pharmacology of the compound? Is it sometimes different? And they're not always the same. Acetaminophen, which we've all heard about, Tylenol, its hepatotoxicity has nothing to do with its CNS effects. Okay, now this is a slide where we've been able to stain a rodent brain with a fluorojade B, which picks out the degenerative neurons. So we can find there are sophisticated ways to look for the target organ. Okay, that's toxicology. So let's talk a little bit about drug development. Here's how most people think about how discoveries are made and translated into the clinic. First, a miracle occurs, right? Then something else happens. It's a black box. It pops out, and then we have a product made in some factory, and then it shows up on the pharmacy shelf. Well, something else happens, okay. All right, so what really happens is discovery is basic research. You've heard the term research and development. Well, those are often frequently two very different lines of activity. Discovery is basically basic research. Development is applied research. One laboratory can make a discovery. It takes a very large organization to do everything that's required to get it commercialized. You got to do the biology, you got to do the pharmacology, you got to do the toxicology, you got to be able to come up with a method to measure it, you got to have a way to make it, you have to have a way to formulate it. All these things are going on. What you're going to hear about, and I'll introduce this a little later, is the path from discovery to approval is now called the critical path. And the type of research that goes on to take something from discovery to approval is called translational research. We want to translate something. The term you'll hear is from lab, from test tube to bedside. And okay, here's another view of this that I got from the Pharmaceutical Manufacturers Research Association. They have what they call pre-discovery, and this might cop out 5,000 to 10,000 compounds, which goes through a screening process to get you into preclinical, where initial toxicology tests and other activities are done to file an IND, an investigational new drug application. All drugs that are sold in the United States must go through clinical trials. All right, and they are divided into three phases. Basically, phase 1 is bioavailability and tolerance testing. Phase 2 is your initial evidence of efficacy. And phase 3 are the well-publicized, double-blind clinical trials to look for efficacy in diseased patients. Okay? And at the end of this, you'll get one approved. That's statistically the way it's working out. For every 5,000 to 10,000 compounds, you'll get about one approved, okay? Right now in the United States, there's probably about 7,000 compounds in various stages of development along here. So maybe we'll be lucky and get 10 new pharmaceuticals out of that. The other thing I wanted to draw your attention to is down here, you can see the amount of time that is estimated for these periods to take. This is the critical path. The amount of time it takes to go from discovery to one FDA approval. Look at the number of patients, the number of volunteers that are frequently involved in these activities. You get into phase 3 clinical trials, some of these will require 5,000, 6,000 patients. Now, take a small hospital like in Ashland, Ohio. There's no way they would ever see 5,000 patients a year, You're talking about several metropolitan hospitals that are involved in this type of activity. Okay, So that's your critical path, and I just wanted to give you some idea where toxicology comes in. We get involved during the preclinical phase where we do what is called IND tox, and then as the drug goes through development, it'll go back and forth between the clinic and the basic laboratory, where we do increasingly larger and more expensive trials. The other area I'm not going to spend too much time on is the chemical side of it. You got to be able to measure it in the bloodstream. You got to be able to look at the clinic, compare the toxicities you're seeing in the preclinical animals with the toxicities you're seeing in the patients. And the only way to do that is on the basis of what you're measuring in the bloodstream. These are staggering starts, okay? This is something that, here's the site that I pulled it out of. The costs are staggering. Okay, let's take one company, AstraZeneca. Between 1997 and 2011, they spent $59 billion, okay? $billion. They got 5 drug approvals, so that cost them roughly $11.8 billion per drug approval. Now, to give you some idea, if you had $11 billion that you could somehow stuff in your basement so that it wasn't getting interest, and you spent $100,000 a day, every day you bought a new house or something, how long would it be before you ran out of money? Anybody want to take a swag? 300 years. Your money would outlive you. Now, if you stuck that in a low-earning interest account, you'd never run out of money. It would just keep. It's an extremely expensive venture. At the same time, drug approvals are not going up. We had a spike back here during the Clinton years, during the Bush years. It tended to bounce along. You take a look at some of these numbers. For all that money, 18 new products a year, 16 new products a year. Okay. Down here, these are the biologics that have been approved. These are the monoclonal antibodies and the large proteins that have a different pathway. Okay, so we're spending a lot of money, and productivity isn't really going up. Okay, the reasons for this, there's no simple answers. Okay, we should have a rational discussion about this, but this is not due to overregulation. I don't want to digress, but political discourse in the United States would lead you to believe there's a simple answer for everything. Well, there isn't. We're a mature country, and our problems are multifactorial. The disease targets are now more difficult to treat. Treating pain is a lot different than trying to cure Alzheimer's disease. With pain, you give the patient medicine, ask him if it feels better, you prick his finger, and if he doesn't feel anything, you've got efficacy. Now think of the problems you have in trying to determine efficacy with Alzheimer's disease. The time it will take, the endpoints that you have to establish. These disease targets will require longer time to deal with. I personally think there's a lack of innovation. I don't know if it's due to discovery or if it's in translational research. I think part of that has to do with direct-to-consumer advertising, all right? Back when I was growing up, drug companies were not allowed to have their commercials on television. It's a dirty little secret that most drug companies spend more on their advertising than they do on their basic research. So it's been diverting resources. There's also been a decreased public funding. I don't know if it's decreased so much as it's remained stagnant. Many scholarly articles have shown us that have gone back and shown how government-funded research, basic research, led to the discoveries that eventually led to new compounds. And to the extent that we're not funding those, there's going to be fewer discoveries in the future. Okay? The fact that it's taking a long period of time has also gotten the attention of the agency. Back in 2004, they launched what they called their critical path initiative because they wanted to decrease that critical path. And then in 2006, they published their scientific priorities organized by categories. For a toxicologist, the one that where we would tend to play would be in better evaluation tools. And what that boiled down to would be disease models, better biomarkers, more use of imaging, and I should also add increased use of in vitro technology. In vitro is the use of cells outside of the body. If you're working with an intact organism, that's called in vivo work. In vitro work is much of what our friends here are talking about. They're working with stem cells outside the body. They could be called ex vivo because they're going to put them back into the body. All right, and if you're interested in all the machinations about the Critical Path Initiative, there's a website here you can check out, or you can just go on Google FDA Critical Path, and it'll take you there. I've used the term biomarkers, and so I wanted to give you some idea of what. Here's the NIH definition of it. It's a biochemical feature or facet that can be used to measure the progress of a disease or the effects of the treatment. Basically, not only do we want to identify a potential target organ, we need a non-invasive diagnostic tool to detect damage and recovery in patients. And I'm talking as a toxicologist. You also want to have a biomarker to watch the progression or the cure of the disease. So hopefully during clinical trials, they have highly specific markers to help them do that. Okay, another place where the government has gotten involved. Okay, now this is the Tox 21 initiative that the FDA signed on to. It was more of an EPA-driven initiative. They weren't so concerned about the time as they were concerned about the sheer volume of chemicals that are still in commerce that have never been analyzed for their toxicity. All right, and if you're interested at all, it's called Tox 21 because hopefully this is what toxicology will look like in the 21st century. I doubt that I'll make it. Maybe some of you will. Anyway, what they want is more in vitro research, more high throughput, developmental models that can more effectively predict how chemicals will affect biological responses, because there's just too many chemicals out there to go through our current routines. So they want increased throughput, more and better data, decreased reliance on animal-based testing. And that could be an opportunity for somebody. Okay, so what's the industrial response been to the increased costs and the Critical Path Initiative? Basically, what they want to do is get more information about their potential drugs in this area here, okay? They want better disease models, increased emphasis on in vitro technologies to give them better information before they even start their preclinical work, so that they have drugs that are more likely to succeed and go through this. Right now, nobody's too anxious to change this paradigm. It will probably change as a result of the technologies we learn right here. Why aren't they, given the time, why aren't they anxious to change this? Well, sometimes it's better the devil you know than you don't know. The regulatory activities change very slowly in the United States. Okay, so these are some of the things they could screen their new drugs for. The buzzword is druggable. What are the compounds, what are the attributes of this compound that will make this a more druggable entity before you even start it down the development trail? And here's a list of some of the things that we look for. Okay, now I'm going to switch gears and start giving you some examples of some of the things that we've been doing to respond to the critical path. One of those, remember, was imaging. So we've developed a way of imaging where the drug goes, and it's called quantitative whole body autoradiography. And this was an animal that was injected with a radioactive compound, and then we can take a picture of it essentially, and then use color color-enhanced imaging to see where the drug went. You know, if the drug doesn't go to an organ, chances are it's not going to affect that organ, all right? And here you can see the different scales that show you that where the highest intensity of the drug is. Right now in this animal, it's still in the liver, in the intestine. What I wanted to draw your attention to is, you know, low concentrations are kind of like this blue. So this particular drug did not enter the blood, did not enter the brain. And this happened to be a gravid rat. And you can see here, these are the fetuses. And the drug did not penetrate there. So we can, with a very few number of rats, determine whether or not this drug has a potential to be a teratogen, the assumption being that something that cannot enter the fetus will not be teratogenic. Anybody know what I mean by teratogenic? I'm so sorry. Cause birth defects, birth deformities. Okay. Okay, here's another application of WQBA, and here we used genetically modified animals. I want to attract your picture down to this picture here. This is a mock-up of an intestinal epithelial cell. All drugs just about have their own transport systems they'll be picked up in the lumen of the gut, transported through, and then another transport system take them into the blood system. Okay. One of the reasons, cancer cells sometimes develop resistance, often develop resistance to therapies, is they grow abnormal amounts of these particular transporters, so that as soon as the drug enters the tumor, it turns around and pumps it right out again. You know, so this is an area of intense research because of the way you can control where a chemical goes. If you can come up with a way to inhibit these and permit the drug to stay in the tumor, then you have a more effective therapy. Here we did some work with knockout mice. These are mice that are genetically engineered to lack some of these transporters. This one lacks MDR1, multi-drug resistant protein 1. This one lacks BCRP, which stands for breast cancer resistant protein, and multi-resistant protein. And what you can see here is that this transporter was very important in excluding this drug from the brain. You can see you get drug penetration there. So here we have a system that can help us determine whether or not something will be toxic to the brain or not. Okay, here's another example of cryoimaging. Okay, and this is a picture of a small mouse fetus. And this one has been genetically engineered so that the smooth muscle will light up under the right kind of light. So now you have, you know, it's much easier to diagnose damage here versus here. So here's an example. This was from an animal that was exposed in utero to a chemical called TCDD. And it caused, here's the normal kidney, here's the normal ureter, and down here is the kidney, but you can see the pelvis is greatly increased in size. The ureter is greatly distended, and it was just much easier to visualize with these. So remember, increased information, target organ identification, more use of imaging. This was actually done by a small startup that's associated with Case Western Reserve. We collaborated with them to do that. The name of the company is BioEnvision. It was started by Dr. Wilson, and these are his collaborators out there. Okay, here's another example of imaging. You know, this is PET scanning, positron emission tomography, done in a rodent. And here what we looked at is actually this particular contrast agent and how it was able to identify a myocardial infarction. You can look at the time scale here, and you don't see any evidence of an infarction here, but you can see where the gaps are developing. And these are different aspects of the heart, you know, sagittal, coronal, etc. So you can see here the time progression that it takes, and you can do this in a single animal now. So these are very powerful technologies that are now coming to us in the preclinical space and could provide opportunities for future research. Okay, you already got a little talk about stem cells. I'm going to talk about another aspect of stem cells, and that's the use of stem cells in toxicology testing. Okay, this is a company that was started just about 10 years ago in Madison, Wisconsin, to use stem cells in toxicology testing. The name of the company is Stamina. There's some advantages and disadvantages associated with the use of stem cells. Most of them have to do with the same things that affect most cell-based cultures. Is, their orientation isn't maintained. The typical occurring adjacent cells aren't there. They're not exposed to normal hormone rhythms. Drug metabolism will be different. The oxygen tension is different. But all that being said, now we have a way of getting very specific human tissues for testing in vitro. And what they do is they grow out cardiomyocytes, hepatocytes, or specific cell types, expose them to the toxin, and then use, and I talked about this, I think it was my second slide, HPLC, mass spec chromatography, to look for biomarkers that are produced by that particular cell type in response to that toxin. Then we can take those to the next step and see if they're actually predictive in vivo. I have no idea, okay. So my last example, and I'll just leave you with this. What I really wanted to do was introduce you to this magazine. If you have a real interest in drug discovery and biomedical research, you might want to check this magazine out. If you missed it at the Giant Eagle newsstand, I'm sure you can buy a subscription to it online or someplace. But this is the next thing, lab on a chip. You take the small amounts of tissue or cells and whatever, you combine it with microfluidics and that extremely sensitive chemical analysis that we can do, and now you have very, very powerful technologies. And in the background here on this cover, they actually show you some of the architecture associated with, you put a little bit of cell here, the fluids you put in, how you take them out, how you analyze them, etc. So, I hope I've given you an introduction to the science of toxicology. I hope I've outlined the pharmaceutical process for you. And I have to admit this is all at about 40,000 feet. So if you have more questions now than you came in with, I probably succeeded. Okay, the role toxicology plays in that process. National initiatives to address that process and examples of entrepreneurship that have taken advantage of this process. This is the company I work for, little old Ashland, Ohio. We have over 1,000 employees located in laboratories around the world. It's hard for people to imagine that right here in little old Ashland, Ohio, there is a world-renowned research and development organization. I've been there, by the way, since 1990, when it was only 85 people. Okay. So, thank you for your kind attention. You've been a wonderful audience, and I hope that we have shed more light than heat on this discussion.